Elisabeth. So we will start as soon as possible since we are already running a little bit late. So it's my pleasure to chair this first session of the morning. And we have three speakers for this first session. And the first one is Hans Berthoud. Hans is coming from the USA, excuse me, from Baton Rouge in Louisiana. He does speak French. He does understand very well French. He spent a lot of time in Geneva before going to the United States. And he will speak about the relationship between hedonic and homeostatic control of feeding behavior, I guess. So Hans, it's your turn.
Bonjour. Unfortunately, that's the only French word probably you will hear in this talk. First, I would like to thank the organizers for giving me this great opportunity to talk to this audience here. We are going to make a pretty big jump now from one end of the food chain to the other end, which you could think of maybe it's the brain or how the humans react to food. So to start with, we really have to understand the fundamentals of energy balance regulation, which is accomplished by a constant sensing of the internal environment, internal milieu, by so-called nutrient sensing. As you can see here, all the peripheral metabolism is sensed. The stream of information is constantly reaching the brain, informing the brain, and the brain makes a decision about both energy intake and energy expenditure, also through the autonomic nervous system, how the metabolism is processed. And this is really based on research that has been done over the last maybe 70 years, which is basically the homeostatic regulator of body weight, which is located mainly in the hypothalamus and the brainstem of the brain. And this is basically responsible for this constant defense of a certain body weight level, as indicated here. We have this defense of a certain level. If we starve an organism, the response is that food intake is increased, energy expenditure decreased. This is a very strong, powerful response, food deprivation, so that the defended level is reached again. And in the other sense, if we have overfeeding, food intake is decreased, energy expenditure reduced. This is usually much weaker than the starvation response. And this is really accomplished or crucial for this process are two populations of neurons. Now, I'm sorry for some of you that maybe are not into the brain, but it's two populations of neurons that are very, very important for the regulation of food intake and the metabolism, which are these AGRP, these red neurons and blue neurons. And together with acting on catabolic and anabolic processes, they basically regulate the fat mass. The feedback signal from fat mass is leptin, which feeds back to these neuron populations. And I just wanted to share with you some recent, very exciting new research in neuroscience, which allows activation of specific populations of neurons. So these, in this case, these red neurons can be selectively activated or inhibited. On the left side, you can see the response if these neurons are inhibited here with a diphtheria toxin receptor that is linked to these neurons. After 2 injections in the brain of this diphtheria toxin, which kills these neurons, these animals, in this case are mice, they stop eating. Everything else is okay with these mice, but they stop eating. They just don't want to eat. On the other hand, we can have obesity on demand. If we activate, stimulate these neurons selectively, we have obesity on demand. These animals eat a lot of food, gain weight. When the stimulation is stopped, they come back. So this is really a very powerful mechanism, which has also been shown in the case of this monogenic obesity, the leptin deficiency. You may have heard about these children that already at a very young age of maybe 5 years, they weigh an enormous amount of body weight. When these people are then given leptin, they decrease food intake and come back to their normal body weight. So if we have this powerful system regulating body weight and adiposity, why is it then that so many people are obese and obesity is increasing? It's a combination of environment and lifestyle through actions on the availability of food, the cost and reward through the food reward, the pleasure of food, the food cues that are present in our environment everywhere through television, the other media. It's the clock. We are awake much longer into the night period. There is stress and mood. There is social habits, and of course the built environment, which to a large extent determines our physical activity. It's a combination really between these environmental and lifestyle factors and the genetic and epigenetic predisposition of that we as individuals have. There is an interaction. Some of us can resist this environment, this obesogenic environment. Some of us cannot resist it. This really is accomplished. These environmental factors interact with the brain really usually in a different parts of the brain than this homeostatic regulator is located. It's mainly the cortex and what is called the limbic system and some other systems. So we can basically say that there are these two areas in the brain, this metabolic domain of the brain, and then this cognitive and emotional brain. And really what I would like to talk with you is about the interaction between these two brain systems in terms of appetite and body weight regulation. So again, we have here in this blue system, which I call the blue system, one important aspect of this homeostatic regulatory system is that all the actions of this system we think are at the unconscious level. These decisions do not reach awareness of an individual. So for example, in the case of a leptin deficient child, you cannot tell that child, don't eat. It's impossible. It's a basic biological drive in the absence of leptin that the child will eat. So in contrast to this blue system on the bottom, I very abbreviated fashion put this cognitive and reward system, the red system, on top of it. And you can see the input are mainly these external stimuli through sight, smell, and taste of food. They interact with different parts of the brain. Basically, what this enables is to lay down the representations of food and also the reward expectancy of these particular foods. These areas are then connected to areas in the brain which have to do with the executive control or decision-making, which then leads to conscious and unconscious actions on energy intake, energy expenditure, and metabolism. So, in a nutshell, the basic function of this system is really to make optimal decisions. So in case you have to choose between an apple and an orange, if you would really, what's going on in the brain if you make that decision? It should be shown in this slide. So this is based on neuroimaging studies. You can see a section here of the cortex, and these two areas here in the orbitofrontal cortex, an area, specialized area in the cortex. In one of these areas, there is a goal value, which is stored in that area, which is the predicted reward from the projected outcome whether you eat the orange or the apple, you have then also a decision value that is basically making the calculation which is at the moment better to eat an apple or an orange. And third, you have to have a prediction error, which is a deviation from the previous reward expectation. So in case the apple was rotten, the next time you eat an apple, you will think twice about eating an apple. You may prefer the orange. So again, what I would like to talk with you is about some of these interactions between what I have painted here as a blue and this red system here, the homeostatic regulator and this emotional or reward system in the brain. And the interactions really go both ways. It's very complicated. You don't follow this diagram. If you want to go, you can read one of the publications. There are many pathways that we can identify, but we just go, I just will mention a few examples of these interactions. One of these examples is the effects that circulating hormones that usually are used for the homeostatic regulator, like leptin, insulin, ghrelin, gut hormones like glucagon, like peptide 1, they seem to interact with other parts in the brain. And this is illustrated in, well, it's also illustrated by the presence of leptin receptors. So again, this leptin is one of these important hormones feeding back from adipose tissue. And the leptin receptor is not just located in the hypothalamus, as you can see on this inset here. It's also located in taste buds. Interesting for you. It's located in the olfactory bulb and mucosa, but it's also located in the cortex and some of the cortex or hippocampus, which is important for the formation of food memories. One example from the mouse literature. So in one study it was found that leptin deficient mice, these are obese mice, they find buried food which is buried under a thick layer of sawdust about 10 times faster than a normal mouse. It's really quite remarkable. When these animals are given leptin back, which they are not producing, this food finding time goes up to the normal level, suggesting, in combination with the knowledge that there are actually leptin receptors in the olfactory mucosa, that leptin plays through the peripheral part of sensing nutrients through the olfactory bulb, has an effect on this sensory function. Similar, there is literature suggesting that this happens with the taste input. I don't have a slide on that. Also, it has been shown here in this study, again, with these children that are leptin deficient, which are always very hungry. In this case, they were looking at pictures of food, and they found that there was a strong activation of activity in one of these areas, this nucleus accumbens, which is one of the major areas responsible for food reward. In the leptin deficient state, there was this strong activation, and this was readily abolished when leptin was injected. In the leptin deficient state, these children even liked food which they normally would never like vegetables and things like that. They were really very strongly motivated to eat such foods. Another example here with ghrelin. You may know ghrelin is one of those hormones secreted from the stomach that stimulates food intake quite strongly. And again, in this study with neuroimaging, also with visual stimuli of food, you can see that in different areas of the brain, here listed amygdala, which is a major emotion center, there is a differential activity whether there was ghrelin infused or not. But the blue system does not only talk to this red system by circulating hormones, it also talks to it by sensory neural pathways through the vagus nerve and maybe through the spinal cord. It has been referred to this constant monitoring of this, for example, is an example of a villus in the small intestine of a rat here, which has labeled vagal terminals. So these are sensory nerve fibers that innervate the tip of these villi in the intestine. Now you have to imagine that the other end of these neurons is in the brain. The vagal sensory neurons have one end in the brain, one end in the small intestine. And so it's not surprising that it has been talked about the gut feeling, because there is this constant flow of information from the inside of our guts to the brain. In fact, this A.D. Craig here has published two very interesting papers talking about how do you feel. He believes actually that this constant flow of interoceptive information from the gut and other organs is part of being aware, that contributes to the human awareness. And this is, for example, supported by observations as shown here, where there was infusion of fatty acids directly in the small intestine, and it was looked at the emotional state of humans. And in this particular paper, it was shown that fatty acid-induced gut-brain signaling attenuated the neural and behavioral effects of sad emotions in humans. You can see here, when fat was infused, this sad emotion that normally was present, or the hunger that was present with the sad emotion, was decreased, and so was the neural activity in several of these critical brain areas. There is also now literature more and more coming showing that the manipulation of the gut microbiome, which should be interesting, I think, to many of you from a nutritional standpoint, with the pro and prebiotics, that in the open field test, that mice behaved differently when they were given lactobacillus. So the title of this paper was "Ingestion of Lactobacillus Regulates Emotional Behavior," and also the neurobiological underpinnings of it, which is in this case the mediation, this transmitter GABA here, which is one of the major neurotransmitters in the brain. And this modulation was really dependent on the vagus nerve. So after vagotomy, this ability was lost here. The same thing here, increased negative emotion in the forced swim test. After vagotomy, this difference was abolished. So there is a whole new field now, which we could label as the microbiota gut-brain axis, by which these microbiota in the small intestine signal primarily through these vagal sensory fibers, but also through the circulation, and change brain function and possibly even brain structure. And there is talk about the search for microbes with psychotropic properties. We also have studied the involvement of the obese state and obesity surgery on the effects of this function of the rat system shown here. So we have, for example, found that when we have rats lick from a spout in this brief access lick test, where rats can lick 10 seconds on each spout, and these are different concentrations of sucrose. Every 10 seconds, it changes here what concentration, and if you do that over time in a rat, you can see that while a normal lean rat that eats normal food, there is this nice increase, as you would assume that the higher the concentration, the more the rat licks. Interestingly, what we found in obese rats, rats that were made obese with a high-fat diet, that this was drastically different. These obese rats didn't really like very much the low concentration here of sucrose, 0.3. Only the higher ones they actually preferred more than the lean rats. And what we did that together with a program where we do gastric bypass surgery in rats and mice. And after gastric bypass surgery, whoops, I did the little mistake. So basically, we restored this function, even a little exaggerated response to these really low concentrations of sucrose. So we restore the lean function in terms of taste sensitivity. And with sucrose taste, we were then interested to see, is this an effect of the surgery itself, or is this an effect because the rats lost all their body weight? And the second one was the case. As you can see, if we actually took these obese rats and instead of doing gastric bypass surgery, we just starved them or food restricted them so that they lost about 100 grams of body weight. This stopped working. Here it is. So they lost about 100 grams of body weight. We also restored this function here. So the conclusion from this is it's not really a specific effect of the surgery. It's because surgery induces weight loss that restores this sensitivity to sucrose. We also have done very similar experiments with fatty taste, and the results are almost the same. We also went one step further, so this brief access test is sort of a test for the general reactivity to taste, but it's not known exactly where in the system this works. So we did another test, which is called the taste reactivity test, where we give a rat just a very small amount, a drop of sucrose of different concentrations, and we actually videotape the oral movements what the rat makes. You know, if you give a cat or a dog a piece of food, you can see they swallow it, and then they show these tongue movements and these tongue protrusions, and you can actually video, the rats do exactly the same thing, and you can videotape that, and you can actually plot the number of these hedonic reactions here, and as you would expect in a lean animal, the higher the sucrose concentration is, the more it responds here with hedonic reactions. Now this function is shifted to the right in an obese rat, as shown in the middle panel. They respond very little to the low concentrations, but more exaggerated to the very high concentrations. And again, after the Roux-en-Y gastric bypass surgery on the right, this is drastically changed, not only from the obese, but even from the lean. They actually prefer now these very low concentrations of sucrose, and they relatively dislike the really high concentrations. And this has been confirmed really in human studies where they used the power of food scale, asking the question, I find myself thinking about food even when I'm not physically hungry, and they showed here that in the obese, this was significantly increased. After bypass surgery, it was decreased. So, in terms of food, the relationship between food reward, hyperphagia, and obesity, we have to be careful to distinguish what is cause and consequence. All the reward functions, like the reward we get from food, can really be produced by pre-existing differences, which are, as I mentioned at the beginning, genetic or epigenetic, developmental programming. It can be the repeated exposure to palatable foods. You know, there is a lot now discussion about whether food can be addictive, and some of the same neural circuits are used for food addiction and for drug addiction. And in drug addiction, it's very clear that repeated exposure to drugs changes by itself how these reward functions work. And the third one can then be the secondary effects of obesity on food reward functions, which can be distinguished only if you use food restriction to verify that. So now I've talked about these bottom-up manipulation or modulation. Let's have a few examples about the top-down modulation of eating by cognitive and emotional processes. So how this red system influences this blue system. One example that has been around quite a while in the literature is conditioned eating. So if we see in the media, if we see on television this advertisement for an enticing food, you know, do we really go and react to that? Well, in human studies, this is not that easy to do. In rats, you can do it very easily. You can basically have a light or tone. You food restrict the rat first, and in the food restricted state, you have a light or tone signal the availability of food in a cup, in a part of their cage. And as you can see here, with the CS+, the conditioned stimulus, which is always paired with the signal present, with the presenting the food, after about 10 sessions, 10 learning sessions, you have a pretty good chance that the animal will go and try to eat that food, almost 90%. If the CS-, if this is not paired, you don't have this association. What you do then is you feed these animals to satiety, so they are metabolically now not hungry anymore, and you sound the tone or the light. And you can show it again and again that when you do that, when you put the CS on, the animal, the rat, goes and eats food. You can do it after an hour again, you turn tone on, the rat goes and eats food. This is conditioned food intake. Has been recently shown in a very nice series of studies by Petrovich here, that a particular connection between the amygdala and parts of the lateral hypothalamus is responsible for this. If she interrupted the connection between these two, as shown on the right, there was no conditioned response anymore. So also research with subliminal stimuli, those stimuli that only are given to you in milliseconds, so that you actually don't remember what you saw, but you act as if you know what you saw. It was shown that expected rewards can energize behavior without the need of the subject's awareness. And another example, which is probably very close to you people here in the Bourgogne, is about wine tasting. Maybe tomorrow afternoon, I was hoping we can do a little bit about that. And so this is really an interesting study, which some of you may have seen. When people have to say how much they like a particular wine, when the wine the label was covered, it was neutral, and the blue wines were the same wine in 2 glasses, and these 2 wines were also the same. It's no surprise that they are liked about equally. This is a little bit less, must not be a Burgundy wine here, I guess. When then there was a price put on the bottle, you can see that immediately people like the one that is more expensive, they like it more. And that has been done over and over again. It also, at the same time, these people were in a magnet to do neuroimaging, and they looked at the activity in this medial orbitofrontal cortex, and you can see that this signal with a $90 wine is not only longer, but also much larger. So it's not just the imagination, you can find a neurological correlate of this idea that we are guided by the price of the wine rather than by the taste. So top-down cognitive processes encoding flavor expectancies are integrated in the medial orbitofrontal cortex. And a quick example from the human literature here with not with neuroimaging is about the memory of food intake. This paper basically shows that manipulation of the memory of a recently consumed meal influences hunger and satiety. And it was done with this incongruous eating, with the magic soup bowl, where the participant has a bowl which he eats from, but the experimenter can siphon off more or less of what he eats. So you eat a certain amount, but the experimenter can fool you how much you actually see that you have eaten. And if you basically here, after 2 hours, Subjects who thought they had consumed a larger soup portion reported less hunger after 2 hours, shown here. And even a day later, they showed increased expected satiation. So you can be fooled by your eyes from what actually went into your stomach. There is interesting papers about it from Suzanne Hicks here, Memory for Reasoned Eating and Energy Regulation, Terry Davison, Memory Inhibition and Energy Regulation. And it was known a while that hippocampal damage, which is really the hippocampus, the center that is responsible for many memory functions, results in reduced satiety and overeating. Maybe one of the most important abilities, of course, is to inhibit inappropriate actions. For example, if we are faced with a breakfast buffet where there are 100 items and they are all very delicious, well, we shouldn't really eat them, all of them, so we have to inhibit our impulses. It's also known as delayed discounting. Because we want to have a healthier life and a better life, we are not doing certain things right now. We inhibit that. And, you know, you can argue how strong that ability is. I mean, there is this yogi here, which apparently said that for 70 years he didn't eat anything, which, of course, we know is impossible, but it is remarkable that people that can go on a hunger strike can actually die because they can just not eat. So you can, under certain circumstances, by your free will, so to speak, you can suppress certain things. But most of us cannot do that. And it has really been shown that this network is located in another area of the brain, which is this dorsolateral prefrontal cortex, which is an inhibition network. It has also been shown that this inhibition network is influenced by both conscious and unconscious information, which is very important. And the last slide here really is to show some of this literature. In this study, people had to press a button press paradigm to make a decision which button to press. And there was an ability to know when the subject became aware of his or her own action, which is indicated here by this red line, aware of choice. And so what you would expect in the motor cortex, you would expect that once the subject is aware of his choice, that then the motor cortex says yes, and you grab the food or whatever, you eat the food. But there are areas, several areas in the brain here, where activity was long before the subject reached awareness here, up to 10 seconds before. The conclusion from this paper was that when the subject's decision reached awareness, it had been influenced by unconscious brain activity by up to 10 seconds. So maybe we are not fully in charge, even if we think it's free will and we make a decision absolutely on our own. Maybe the way we got that decision was completely at the subconscious level, reached at the subconscious level. So let me conclude then. Just 3 quick points. The neural systems mediating food intake and energy balance are complex, distributed, and redundant. And the only way we can really get this system under control in terms of treatment is with combination therapies, probably. We have to use both behavior modification and drugs. We have to probably attack both the homeostatic and hedonic mechanisms at the same time. We have to probably attack both energy intake and energy expenditure at the same time. And I would, being here with you, I would say that smart nutrition is, of course, central to all of this. Very early, I mentioned that although very powerful in defending a lower level of body weight, the homeostatic regulator appears to be overwhelmed by an environment of plenty in susceptible individuals. So I think this would be the place to ask that the government, industry, scientists have to come together to solve this problem. This is an enormous problem, and we really have to talk with each other to solve this. And I think that the rules of interaction between these two systems, the blue and the red, are likely to be important for understanding the treatment of common obesity. Thank you very much.
Thank you very much, Hans. It was a very nice talk and a large overview of everything which controls the feeding behavior in humans, and a quite complex system, as you said. So we have time for 2 or 3 questions, if any in the assistants. So maybe I can start with one. You mentioned in one of your slides food addiction. And as you know, there is quite controversy about the term of food addiction or not. So can you develop a little bit or give us your opinion on that system?
Yes. Well, I think that the best reason to say it's not the same as drug addiction is because we have to eat food every day. And it's simply that. And drugs, of course, we don't have to take. That makes it really very different. And I think even the palatable food has a role to play here. It has a role to play to eat very different foods with different nutritional values. It is possible that with these very refined foods that we have, maybe a lot of the pure sugar and things like that, which probably, even 50 years ago we had much less, is probably not the right thing to do. That with sugar, you can really, there are interesting experiments from actually some of your countrymen in Bordeaux. They have shown that sugar is stronger than cocaine in terms of attracting a rat's or a mouse's interest. And so obviously these stimuli are very strong in its pure form. So I would suggest that nutrients are best if they are mixed, if they are not pure, if they are not refined, if they are mixed. So anyway, we could go on, and this is a
sure
discussion.
So maybe I have a last question before moving to the next speaker. You mentioned also the relationship between microbiota, the gut and the brain axis, and you gave an example only on the emotional consequences of that. Are there also data showing that there are metabolic consequences directly from the gut to microbiota to the gut and the brain?
Right. This is an evolving field. Again, it seems to change anxiety level under certain conditions and so on. But it also has been shown there are these, and I think one of the speakers may be touching on some of this with these microbiota transplants and the effects. So it has been shown that microbiota from an obese person is transplanted or to a lean one, or in mice it has also been done, that then they actually take partly on the phenotype of an obese also. What the big question really is in this area is how the signaling goes. How do the microbiota signal to the brain? What kind of chemicals, what kind of circulating factors and the nervous system is involved in this. And this is really an very exciting evolving area.
Okay, thank you, Hans. So we can applaud Hans once again. Thank you very much.